everyone, welcome to Biomics Hub. Today in this video we are going to discuss another one topic or another one technique, that is the ELISPOT. This ELISPOT is also known as enzyme linked immune absorbent spot. So what is the function of this type of techniques? Suppose we are having a cell and we want to know that whether any cytokine is secreted in or not. So for that we can use such type of techniques. And this ELISPOT is used for the detection of the cytokine synthesis in specifically one cell or a specifically single cell. So let us see what are the things required for this technique to be performed. The first thing which is required is a cell. So which kind of cell? Suppose we are targeting some of the cell and we want to know that whether there is production of the cytokine from that cell or not, and specifically one cell. So that cell we can take in this technique. The second thing what we require is the stimulus. So if we are having a cell, some cells will be present who can secrete the cytokine. Whenever they want, they can secrete the cytokines. But in some cases, when the cell is present, whenever it will get a stimulus for the secretion of the cytokine, then only it can release the cytokine. That is the reason why we are using the stimulus. Next is a capture antibody. This capture antibody which we will use, it is specific to cytokine. Next is the cytokine which will be released by this cell. Next is a detection antibody. So we are performing this in a tube. So we cannot visualize everything. They are in the solution form. They are in the volume. They are in the solution. So for identifying, we need some of the detection antibody. Next is a substrate. Whenever we are using the anti-detection antibody, we are using the enzyme. The antibody is enzyme linked. But whenever the enzyme is present, how can we visualize or how can we know that there is some changes? For that reason, we are using some of the substrate which is specific to the enzyme. So one by one, we will discuss how it is performed. So here in the first well, we can see that there is antibody, capture antibody present, which is attached to the microtiter well. Here we are having the capture antibody, and this antibody is specifically can bind with the cytokines. The next thing which is added here is a cell. We can see the picture here. This is a picture of the cell. The cell has been added into this tube. So now the cell which is being given, it can produce a cytokine, or when it will be provided some stimulus, it can produce a cytokine. So here there is a stimuli given who will stimulate the cell to produce the cytokine. In the second tube, you can see here that there is production of cytokines here after the cell has been attached with the stimuli. Here the cell is present and we are here having a stimuli which produces a cytokine. And the cytokine, as it is having one of the antibody which is specific to the cytokine, this cytokine is working as an antigen and the capture antibody is capturing the cytokine. Now, there is the production of cytokine and antibody has already captured it, but how can we detect that? For the detection, we need another antibody, that is a detection antibody. This is a detection antibody. Here what happens, the antigen which is being captured by the capture antibody in other epitope, in other area of the antigen, the detection antibody can come and bind. The next thing which is performed is, in the detection antibody, there is a enzyme link. And for this enzyme, there is a specific substrate present. So we can add the substrate and here we can see the color change. Okay. When the substrate is added, there will be a production of some of the color. And in case of ELISPOT, when the color is produced, it will be seen in the form of a spot. Here in the fifth, you can see that it is giving a spot. In this way, ELISPOT can be visualized if the cytokine is present in a single cell. Let us see with this other diagram. So here we can see that there is a capture antibody present. So this capture antibody is specific to the cytokine, and after that we have to code this part with BSA. It may be bovine serum albumin. Why we are coating the other surfaces? To avoid the false detection. We are avoiding the surface which are free. The next thing which is added here is, it is the cell, and the cell is secreting the cytokine. Here no stimulus has been given, but we can give the stimulus because all the cell cannot secrete the cytokine. Whenever there is a stimulus given, they can secrete the cytokine if they cannot produce alone. So here, after this, it is being washed, the cells has been washed, only the cytokine, which is working as an antigen, is present here. The next thing which is added here is antibody. This is a secondary antibody, or we can also tell it as that it is a it is not a capture antibody, it is a detection antibody. What happens in the detection antibody? Here there is biotin attached. Okay, so there is a substrate known as biotin, and this biotin can bind with avidin. So in case of antibody, here biotin has been added. Okay, now the enzyme which will be added, this enzyme will be coated with avidin. So this enzyme and avidin can bind with this antibody which is coated with biotin. Here we can see that this secondary antibody is been linked with the biotin. Now in the next step, the enzyme specific to this is added, and this enzyme which is added, it is coated with avidin because it can bind with the antibody, as the antibody is having the biotin. The next, according to this enzyme, one substrate is given to this so that it can give some of the color. There will be a color change if it is reacting. So whenever we are giving a substrate, it will bind with this, enzyme, and it will produce a color. That is the reason it will give some of the spots, which is also known as ELISPOT. This was all about the ELISPOT technique. Thank you for watching this video till the end, and if you like this video, please do like, share and subscribe. Thank you.